Welcome to Cryo-Talk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific, featuring conversations between your host, Eva Amson, and experts in the field of cryo-electron microscopy.
Today on Cryo-Talk, we're joined by Gökhan Tolun, Associate Professor in the School of Chemistry and Molecular Bioscience at the University of Wollongong in Australia, and Research Group Leader at the Molecular Horizons Research Institute. He talks about research funding challenges in different countries.
When I was in Turkey, we would use a lot of handmade equipment, for example, like plexiglass glued to make containers for running gels and things like that.
How Molecular Horizons incorporated a custom-built microscope facility.
When they were looking for a place to build it, they had to find on campus the area where the electrical interference was lowest.
And what he does when he finds some spare time.
I still have my archery equipment here. So there's an archery group here that I every so often go there and shoot with them on weekends.
All in this episode of Cryo-Talk. Hi, and welcome to Cryo-Talk. I'm Eva Amson, and I'm here today with Gökhan Tolun. Gökhan is Associate Professor in the School of Chemistry and Molecular Bioscience at the University of Wollongong in Australia and Research Group Leader at the Molecular Horizons Research Institute. So how are you today?
Fine, and you?
I'm good. So, it's morning for me and it's evening for you in Australia.
Yes, it is evening.
Now, the first question we tend to ask our guests is, can you tell us a little bit about your career so far?
Yeah. So I'm originally from Turkey, so I'm originally Turkish. So I started my career back in Turkey. So I attended undergrad there to Department of Biology. And then I actually started my graduate studies there. So at the time, doing a master's was a prerequisite for a PhD. So I have a master's in biotechnology. And then I actually started my PhD back in Turkey and did a couple of years there. And then meanwhile, we were applying for PhD positions in the US, and we got an offer. So when I say we, me and my wife, she's also a scientist. So what we did, we just left our program and then went to the US and started from scratch, another PhD. So I got my PhD from University of Miami in Florida, and then moved to North Carolina, to University of North Carolina in Chapel Hill for my first postdoc, where I started with electron microscopy. So that was more classical EM. We were doing what is called a shadow casting EM. So you mount your molecules. You still work with purified molecules like we do in cryo-EM, as we are going to talk about. But in this method, although you are working with purified samples mounted onto the EM grids, we are actually evaporating metal onto the sample. So that's why it's called shadowing. So we shadow cast the molecules by covering them by evaporating metal onto them. And also negative staining, which is a pretty common technique in electron microscopy. So after that, I moved on to learning cryo-EM. And for that, I moved to National Institutes of Health in Bethesda in Maryland, in US still. And then I was there for more than a couple of years. And then I moved to another institute within the NIH to continue my postdoctoral training to Frederick this time, NCI, National Cancer Institute under NIH. And then after that, I got my first independent position in Australia, close to the beginning of 2018. And came here and established my independent research group with the help of my first postdoc, Jody Brewster, who is moving to an industry position tomorrow, actually.
Oh.
So I think in a nutshell, that's been my career so far.
So it's been very international. And
Yes,
before,
three continents,
yeah, yeah.
three continents, three countries.
So what have you found the differences to be in the research environment between Australia, the US, and even Turkey, where you did part of your first research?
Yeah. So, of course, there are some funding considerations
Mm,
and limitations. So, Turkey, the science funding in Turkey is a lot more limited than in US and Australia. And of course, the most funding is available in the US, I would say,
mm,
than Australia. But still, people, of course, do the best they can everywhere. Scientists are scientists. They're enthusiastic about the questions they want to answer, and then they do their best. So when I was in Turkey, we would use a lot of handmade equipment, for example, like plexiglass glued to make containers for running gels and things like that, or homemade instruments for doing electroporation to transform bacteria and things like that. So yeah, even though funding is limited, people find ways to do science, which is
yeah,
great. But I wish there was more funding, of course. So and
yeah,
then US, of course, is a big country and doing well financially. So because of that, there is of course more funding for science. So Australia is also pretty good. The issue with all of these is the percentage of funding compared to the number of available scientists who would like to do research. So I wish there was more resources being put by the federal governments everywhere, all over the world, for doing more science. So I was very lucky to be awarded an HMRC Ideas Grant a year after coming to Australia. So that was more than 600,000 Australian dollars. It is sort of equivalent to the NIH R01 grants, the major grants. So, but the success rate was 11%.
Mm,
So that means
yeah,
out of the 10 people applying for the grants, 9 researchers are not funded, right?
yeah.
So we
Was it the first
definitely,
time you applied?
yes, it was the first time I
Oh, you
applied.
got really lucky. Great.
Yes, I know. So, and then I'm also a part of another grant here. It's called ARC, Australian Research Council. So that is equivalent to the National Science Foundation, NSF, in the US. So I'm
Mhm.
also a part of that grant together with another lead academic. So yeah, that's the difference in funding. As the uh, scientific, what is the word? Mm, like people goes, of course, Australia is much smaller, right?
Mm,
Like 1/10 of US. So of
yeah,
course, when you attend a meeting in the US, and also people tend to go more to US like internationally for
yeah.
meetings. So I get to see a lot more people and then connect with a lot more people when you attend meetings in the US. But
Mm,
again, Australia, although being remote, it's not too bad. So,
yeah. Yeah,
but
I was going to
Australia is,
ask you about
yeah,
international meetings actually, because most international conferences tend to be in the US or in Europe, and you're quite far removed from it now. So do you really have to decide like, oh, is this conference going to be worth it? Because it's a long journey.
it's not only long journey. As I said, the funding is limited, right?
Yeah.
So I
Yeah.
wouldn't mind the long journeys for attending good meetings. So usually it comes down to, since Australia is so remote, for going to any meeting outside of Australia, you need to fly long distances,
Mm,
which turns out being expensive.
mm,
So
yeah.
usually, again, funding, uh, in addition to time, becomes a limiting factor sometimes.
Mm.
But yes, I do miss going to those meetings in the US and meeting all those people.
Mm. Yeah. And what's your research focusing on at the moment?
So my research is studying pathways in, I would say, generally nucleic acid metabolism. So studying DNA, RNA in general. So more specifically, my group is interested in studying a reaction we call single-strand annealing, homologous DNA recombination. So this reaction takes place pretty much in all life. So it's a very conserved mechanism of stitching DNA together when it's broken. So it's one of the DNA repair pathways. So what happens is that when two DNA molecules are broken with some homology between them, these systems can chew them up and then stitch them back together to make an intact piece of DNA. And this is, as I said, found in organisms as simple as bacteriophages. So bacteriophages are viruses that infect bacteria. So they are, that low in the evolution. And then also as humans. So this reaction takes place, like I said, in all organisms. So that's what we are studying. And you may think that for a reaction that is so conserved and found in all life, we would know a lot about it, right? Turns out we don't know a lot about it. So my group is focusing on studying that. And to do that, we are using two model systems mainly. So one is the bacteriophages that I mentioned. So we are studying a few different bacteriophages, and then also herpes viruses. So as the human pathogens. So mostly viral DNA recombination. But as I said, it's not just limited to that. For example, one of the collaboration projects that we published relatively recently was studying RNA metabolism, so transcription. And that was also an interesting project where we learned how the machinery that synthesizes the RNA message from DNA template when it gets stuck, it's a problem, because then it becomes a roadblock on the DNA. So something needs to remove that, take it off. So otherwise, another transcription machinery that comes bumps into that and it cannot go through. Or even worse, if the DNA replication machinery comes to that, then it's a disaster. So the cells developed ways of rescuing these stalled transcription complexes. So we studied one from a bacterium and showed how a factor called another protein that interacts with the RNA polymerase synthesizing the RNA binds to this complex. And it actually looks like, we used to call it like a gorilla or Hulk. It looks like it has two arms and a head and a body. So what it does is it puts its right arm all the way into RNA polymerase, into the active site, to kick out the DNA and the RNA from there. And with its left arm or hand, it grabs the RNA polymerase and pushes it to open it up so that, it can be removed from the DNA template that it's stuck on. So we showed that by using cryo-EM.
Yeah. That was going to be my next question, actually. So were you using cryo-EM for that? Yeah.
Yes, we used cryo-EM for that, and we are mainly using cryo-EM also to study these viral DNA recombination complexes by determining the three-dimensional structures of these machinery. I call them bio-nanomachines. So they are these fascinating complexes that carry out these processes. Yeah.
And how many people are in your research group now? Because your group is about 5 years old, I think.
Yeah. So as I said, I came here beginning of 2018. So I was actually looking at the numbers recently, so I jotted them down here. So far, my group had trained 2 postdocs, 6 PhD students, 10 honors students, and 7 undergraduate students. So, some of them, of course, like the undergrads and honors students, they come and go, stay for a semester or a year. But I still have the PhD students, and one of them is just submitted his thesis, so he's finishing.
Yay.
And
Great.
like as I said, one of the postdocs just moved to a company. So yeah, training lots
Great.
of scientists.
Yeah. Do you have any lab traditions?
Lab traditions, we
Yeah.
have lab lunches.
Nice.
So like every month or so, we just go have lunch together, and then usually once or twice a year we go somewhere outside the campus.
Mhm.
Again, usually it's lunch, or if we can manage it, sometimes we meet at our place. So I just invite all of my lab to our house,
Mm.
and then just have a nice day.
Nice. Great.
Yeah.
And so can you tell me a little bit about Molecular Horizons? Because I read a bit about it and it sounds like it's going to do some great things.
Well, hopefully we already have been doing some great things.
Yes.
So Molecular Horizons is the largest investment by University of Wollongong. So it was more than $80 million to build that from scratch. And the interesting thing about it is that it was designed from the ground up for cryo-EM, mainly for cryo-EM. Well, housing, let's say, the cryo-EM microscopes, electron microscopes. So when they were looking for a place to build it, they had to find on campus the area where the electrical interference was lowest.
Oh,
So they said,
wow.
okay, we can only put the microscopes here. So they had to build it there. And then when they were building the building, they didn't use ferrous materials, for example. So it's very high tech. So they have these special polymers in the concrete instead of the steel bars, and instead of the steel ties, they used plastic ziplocs. So sometimes, when I give a talk, I joke about, oh, this is a building held up by zip ties, plastic zip ties. So it's a very high-tech building and designed, like I said, from the ground up to provide the best environment for these very sensitive cryo-electron microscopes. So therefore, our microscopes are performing quite well.
Yeah. And so were the microscopes, did they have to be moved from one part of the campus to another, or are they mostly new?
So when I came, the medium and let's say microscope Arctica, Talos Arctica, that was on campus, but it was in another building that was in a room that was retrofitted. So it was performing okay, but it wasn't, doing its best. So they had to install some active field cancellation systems, and the room temperature wasn't as strictly controlled. There weren't any dampeners in the room walls. So it had to be moved from that building to our building. And our high-end microscope, the big microscope Titan Krios, that was located in the electron microscopy facility of ANSTO. So that's about, I don't know, like 45 minutes drive from Wollongong. So that had to be, sort of taken apart and then shipped, moved to Wollongong campus and put together again. So that was a stressful time. But all is working. So our facility is run by Dr. James Bauer, who is also from America. So he came here from University of California, San Diego. And he's doing a great job keeping our facility running tip-top shape.
Great. Yeah, I think most places don't have such a special built place for cryo-EM, and everyone is just kind of making do with whatever space they have. So
yes, that's
amazing.
that's the special thing about Molecular Horizons. Absolutely, you're absolutely right.
And what do you do when you're not working? Do you have any hobbies?
I have some hobbies. The thing is, when you're a teaching and research academic, there isn't much time to do anything else. Really, too busy.
What would you like to do if you had
what
time?
what-
Let's put it that way.
what I used to do, let's say, when I had time to do other things than science. So I'm an amateur photographer. So I'm not only taking pictures of molecules. I actually love taking, photos, mostly nature, animals, things like that. So I do photography when I have time. And I grew up, in these like 70s, 80s. So I am a computer generation. I'm in the computer generation. So I actually love playing video games. But again, uh, unfortunately, they take up too much time. So
they take up a lot of time sometimes.
so again, not much of that, unfortunately. I used to do when I was in at NIH in Maryland. I started doing archery.
Oh, great.
So I was doing archery, and then I still have my archery equipment here. So there's an archery group here that I every so often go there and shoot with them on weekends. But again, it's usually a few times a year, unfortunately.
Ooh.
And I love snorkeling, swimming, like lap swimming or snorkeling, and biking and things like that.
Yeah, well, you are close to the coast, so you have a place to go snorkeling, I guess. And do you ever combine your hobbies? Have you taken photos of archery? Or
I did a little bit of that. Yes. You end up with some cool shots sometimes, like catching arrows in flight in the air and things like that.
Yeah, I can imagine. I was trying to imagine archery in my head when you were talking about it. That's why I was asking if you ever photographed it, because it does look interesting. And do you like to read? Do you have any book recommendations for our listeners?
Yeah, and it's the same thing. I used to read when I was younger.
Of course.
So when I was a little kid, I started reading things like Jules Verne. So I think I read all books by Jules Verne when I was a kid, and then I started reading Clive Cussler, more action type, light novels, and Tom Clancy. I like Tom Clancy's books. And when you asked me, I was thinking, oh, what else did I like when I was reading more? And I remember this book called Shibumi from the author Trevanian. So that was an interesting book, for example. So I would recommend these.
Cool. We'll have to check that out. And what about things on screen, film or TV? Do you have anything that you've seen recently that you enjoyed?
Yeah, we usually, we used to go to movies, but although we still do every so often, most of the time, we are just streaming movies at home now.
Yeah.
So, on weekends, usually we pick something instead of going to Blockbuster and picking up a VHS. So it depends on our mood, but, doing a highly stressful job, we usually go for lighter things when we have time to relax a little bit. So usually we go for comedies, light things, like action movies. And again, when you asked, I was thinking about, what other movies, like, which are not so light that I liked. And one thing that came into my mind was Minority Report. So it's still sort of an actionish movie, but I think it had an interesting message, and it was I thought it was well done futuristic movie.
Yeah.
So yeah.
Do you like to cook? Are you ever in the kitchen? No time for that either.
Not really. So usually my wife is the one who cooks when we cook. I'm not much of a cook. I mean, I do like my best meal is breakfast. So like on weekends when we are preparing breakfast, I'm the usual one who cooks the eggs and things like that. But other than that, I'm not really. I don't think I can call myself a cook. Though
Well,
when I was single back in Turkey, attending the university, I used to cook, so I can actually cook a good number of Turkish dishes.
nice. And breakfast, they say, is the most important meal of the day, so you've got that covered. And do you have any favorite music? Do you listen to music?
I do. I have an hour commute one way,
Mm.
so 2 hours every day. I have an opportunity to listen to some music. Uh, Again, like with the movies, it depends on my mood, what I listen to. I listen to all sorts of music, so it's not like I like this, I don't like that. I love
Mhm.
pretty much all genres, but yeah, it depends on my mood. So I also, every so often feel like listening to music from my time, like 80s, 90s,
Mhm.
like I don't know,
Everyone loves the 80s and 90s music, so yes.
like Sting, Queen, things like
Mm.
that. But I also listen to much newer ones, like I don't know, like Timmy Trumpet.
Mhm. And this is a question I love asking people. If you were not a scientist, what would you be? What would your other alternative life career be?
So interesting question, because I did think of this when I was looking for an independent position. So these independent positions are not easy to find or easy to get. So you think about what would be plan B if this doesn't
Mhm.
work out. So since as I said, I do photography, that was of course one of the things I thought of,
Yeah.
because these, especially like wedding photographers, they do earn quite nice money, I heard. So if I was gonna do something just for money, then being a photographer probably would be an alternative. And I'm also fairly good with computers, so I can actually do a lot of the IT jobs probably. So again, that would probably make a decent amount of money.
If you hadn't gotten your grants, you'd had some backup. And my last question is, do you have any advice that you tend to give to your students, or maybe advice that you've gotten that you'd like to share with our listeners? Anything career-wise?
uh, Anything career wise, science, if you decide to do something, it has to be something you love. So you can't do science if you don't absolutely love it. It's not easy, and as I said, like funding is getting lower and lower, unfortunately.
Mhm.
So and it's as I said, the positions are not that available. Not many positions are available, so you really need to uh, love what you do. For example, when I was back in the US, uh, first of all, as I said, we sacrificed a lot, right? Like I left my family and friends and went to the US to do
Mm.
PhD, and then moved on again. We left everything there, our home, all of pretty much our possessions and our friends, unfortunately, and moved to Australia now. So it takes some sacrifice to do that. And in addition to these, when I was at the NIH for about 5 years, my wife was in North Carolina still, because she had a pretty good job
Mm.
there. So, and initially I was planning to do only a postdoc for a couple of years.
Yeah.
So we said, okay, so we can manage for a couple of years, and it ended up being 3 years, and then 4 years, and then 5 years. So
Mm.
we actually lived in two different states for five
Yeah,
years,
wow.
seeing each
Yeah.
other like once a month or so for a weekend.
Yeah,
So
and
that,
it's not too far, but I guess, yeah, if you have to do it often, for just a few days, it becomes a big
yeah,
big
it
trip.
was like 5 or 6 hour drive. So it's not
Yeah.
that close. We usually flew, but yeah, that
Mm.
was a sacrifice, for example, right?
Mm.
So since she is also a scientist, we have what people call the two-body problem. So
Yeah.
we both need science jobs that are not easy to find. So sometimes it requires some sacrifice.
Yeah, so you have to love science to make the sacrifice. That's kind of the lesson.
All the sacrifices it requires, yeah.
Yeah.
So when I said difficult to find science jobs, I was referring to our specific case with me and my wife, because we are advanced in our careers and very specialized in what we do, and we also require specific instrumentation and equipment to be able to do what we do. But getting a PhD gives you lots of transferable skills in addition to the field-specific expertise you gain during your PhD. So some of these are well known, like critical thinking, including critical reading and ability to synthesize information, so you can absorb a lot of knowledge and then think about how to distill the most important part of it and put it in whatever context it needs to be presented in. So there are many positions available in addition to academic positions for someone with a PhD. For example, you can work in pharmaceutical companies, in drug development, or as a product manager. Or if on top of your PhD, if you get an MBA degree, for example, you can become a business development manager. And biotech companies, other than the pharmaceutical companies, including startups, hire scientists. And in addition to the industrial positions, there are also government positions available. You can become a grant officer, for example, scientific advisor. There are also companies that work on scientific communication, not like yours. And some of them are more specific to scientific writing. There are patent-related positions, since science involved in industry usually involves patent applications and acquiring patents. And you can also work for if you prefer not to work for a company or a government, you can work for a non-profit organization. So there are lots of options other than academia. But of course, I am an academic, so from my perspective, someone like me who's doing novel research, at the moment, when we have made the discovery, we are the only one in the world that knows that specific piece of information. So no one ever lived, or no one living in the history of the world ever knew that knowledge, right? So then we get to tell everyone else by publishing a peer-reviewed paper, a scientific article. We disseminate that information to everyone else, and from that point on in human history, that is now something known, which was before we discovered that was an unknown. So that's the moment we pushed the envelope and expanded our knowledge, and that's how we scientific researchers make an impact on society, make a contribution to the society. And I think it's pretty cool. So how awesome is that? So all in all, I love what I do, and despite all of its challenges and sacrifices it requires, I cannot think of I don't think I can do anything else. So thank you for your time and for giving me an opportunity for this interview.
Well, thank you so much, Gökhan. That brings us to the end of our episode today. And thank you, everyone, for listening or watching Cryo-Talk.
Thank you for listening to Cryo-Talk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific. To view all audio and video recordings from this series, please visit bitesizebio.com/Cryo-Talk.